Dear students, welcome to online classes of biology. So, in the previous class, we were studying about various topics like restriction endonucleases, and also we have seen the gel electrophoresis through the live class. So, today we are going to study about a new topic, that is cloning vectors. Okay, so I have already explained you the gene, foreign gene should be cut and then it should be joined with some host cells. So for that there should be some mediators, okay, vectors that is able to take the foreign gene and deliver into the host cell, isn't it? So the usually used cloning vectors are plasmids and bacteriophages. Do you know what are plasmids and bacteriophages? Bacteriophages are viruses which attacks bacteria, and plasmids are what? The extra chromosomal DNA that are found in certain bacteria, isn't it? So what is the speciality of this? They are having the ability to replicate within the bacterial cells independent of control of chromosomal DNA. Means they are having the capacity to divide themselves, okay, without any direction from the chromosomal DNA. Understood? So they are extra chromosomal DNA, isn't it? In addition to the chromosomal DNA, some bacteria are having plasmids, and what bacteriophages are, what viruses only, they are also attacking the bacterial cells. Okay, so when you compare this bacteriophages and plasmids, bacteriophages are having high number per cell. Okay, means a high the number per cell, so they are what can be done, the introduced gene, okay, can be produced large in number. The copy numbers of their genome will be very high when compared to plasmids. Plasmids may be what, one or two in a cell, or can be 15 to 100 copies, but bacteriophages, it is not like that. They can make a number of copies. So if you link a gene of our interest into bacteriophages, okay, the number, the copy number produced by bacteriophages will be more than the plasmids, isn't it? Then the vectors, now what we are using in biotechnological methods, they are engineered, means biotechnologically they are modified in such a way that they can easily, okay, means foreign DNA can be easily linked to it, and also we can select the recombinants from the non-recombinant. That kind of plasmids or vectors are designed now. Understood? So easily we can add, and also we can what, select the recombinants from the non-recombinants. Okay, recombinants means what? Already see, even though we do all this process for the cutting of DNA, foreign DNA, then what, we are adding this to the vectors. Okay, it is not sure that all the vectors will successfully pick up the foreign DNA. Understood? So what we have to do is, we have to what we have to select which is already got the foreign DNA, successfully added the foreign DNA, and which is not. So this is also very important. Okay, so the vectors nowadays used are designed in such a way that they what, help easy linking of the foreign DNA, and also they help to select recombinants from the non-recombinants. Non-recombinants and recombinants, it's very clear now. Recombinants means which is already successful, which is having what, the foreign DNA successfully added in it. Non-recombinance means the that our procedure or our experiment was not successful. The no recombination, no adding of foreign gene happened in that. So we have to select the recombinants from non-recombinants. That is also an important step. So the vectors designed nowadays are doing both these functions. Okay, then if you want to take a what, vector for this our experiments, they should have some features. So what are the features that, let's see. Okay, so the first is the origin of replication. Okay, so origin of replication means it's a sequence, okay, or a piece of DNA to means which is needed for the replications to start. Okay, so the replication, I told you this when we were studying replication, isn't it? Origin of replication through from there only, or replication starts in a DNA. Okay, so it's a particular sequence of DNA. Okay, from where the replication starts. Okay, so this in addition to that, this sequence is also responsible for controlling the copy number. How many numbers are formed? Okay, so this should be there. Okay, a vector should have a origin of replication. This is the sequence of DNA from where the replication starts. Okay, so that what the gene which we are added, that can also be getting replicated. Okay, then what it also controls the copy number. How many numbers of copies are produced, okay, of the gene of our interest, that is also controlled by the origin of replication. Understood? So if you want a number of copies, what for we are making this copies? Because see, once the gene is isolated, if you add this into a vector, okay, as the vector replicate, you will get a number of copies. Otherwise, again you have to do all the steps from the beginning. Okay, cutting, isolating, so it's not wise, isn't it? So once the gene is produced, we can get a number of copies of that if you align, or if you add this to a vector with what high means vector with a origin of replication which supports number of copies. Understood? Then next what we need is a selectable marker. Okay, so selectable marker means which helps in identifying, okay, and eliminating non-transformants. Okay, what is non-transformants? I told, even though we do this all experiments, okay, it is not sure that in all the DNA the genes are successfully added. Okay, gene of our interest is successfully added. Okay, there can be differences. So in this case, okay, we have to select the gene of means the recombinance, means where the successful what growth is happened, that we have to select during transformation. Okay, so what is transformation? It's a process through which, okay, a piece of DNA is introduced into a host bacterium. Okay, it can be a vector also. So we have to identify which all the the plasmids, or which all the vectors or host, okay, when it is introduced into the host, which all got transformation, okay, which all got the new DNA fragment, and which all not. Okay, so this is what transformant. So we have to identify the transformants from the non-transformant, and for that we need a selectable marker. Okay, so this selectable marker helps in identifying and eliminating non-transformants from what the other one, and selectively permitting the growth of the transformants. Okay, the transformants only will be allowed to grow, and the non-transformants will not be able to grow in the medium. Understood? So that kind of gene sequences are needed. Understood? So usually the genes which are coding, okay, or encoding resistance to antibiotics will be used mainly as a selectable marker. For example, the genes which are resistant to antibiotics like ampicillin, chloramphenicol, okay, tetracycline, kanamycin, etc. Okay, genes, understood now? This selector vector should have some genes which are giving what? Resistance to antibiotics. Okay, and these are considered as what? Selectable markers for E. coli. Okay, so normal E. coli cells cannot grow in this kind of antibiotic containing medium. Only those which are what? Not able to, means which are having this gene which is having resistant to this kind of antibiotics. Okay, they are used as a selectable marker. Means when you grow this bacterium in a medium containing this kind of antibiotics, the transformants, okay, that is which are having the selectable marker with what antibiotic resistant quality only can grow in that, and the others which are not got the genes, okay, or which are not having the antibiotic resistant gene in them, they will die. So we can just collect the ones which are able to live in a medium containing antibiotic resistance, antibiotic, and that we can ensure that these are transformant ones. Understood? So selectable marker is used for what? For identifying non-transformants and transformants. Okay, and transformants will be allowed to grow in the medium. So what they are sequences of genes only, gene sequences which usually provide what? Antibiotic resistance. Understood? So what we are learning, the qualities of a vector. So a vector should have a origin of replication, vector should have a selectable marker. Okay, then next is what? A vector should have cloning sites. Okay, that is in order to link the DNA which we took from other organism, that should be linked to a particular vector, isn't it? So to link that alien DNA, the vector should have a recognition site, and it should be preferably one. Okay, understand? Or very few, or one is more what preferable. Why? Because if there are more recognition sites within the vector, what will happen? In everywhere it will get the foreign gene. We are just adding the foreign gene, isn't it, into the what this vector. Understood? So they can go and add into anywhere, and it will create a lot of confusion. Understood? So to avoid this, okay, the cloning vector should have preferably a single recognition site. Okay, so what happens? This also have asked what will happen if a vector has more than one recognition sites. What will happen? It will generate several fragments, and which will complicate the gene cloning. Okay, so the ligation or the adding of the alien DNA is done in a restriction site present in one of the two antibiotic resistance genes. For example, foreign DNA, okay, at BAM H1 site, you can see this is a vector. This is the diagram of a vector PBR 322, okay, PBR 322, okay, and it has having different restriction sites like HIND3, EcoR1, okay, we learned all this. BAM H1, okay, SAL1, PVU2, PST1, CLA1, all these are what? Restriction sites. Okay. Now, the ligation of the means the ligation of foreign DNA, okay, is done at the BAM H1 site of tetracycline resistance gene. So here you can see there are two antibiotic resistant sites. What are they? Tetracycline sites, okay, this much, and this is the ampicillin resistant site. Okay, so in this tetracycline resistant site, okay, genes, okay, you can see in the BAM H1 site the foreign gene can be added. Okay, so the recombinant plasmids will lose the tetracycline resistance. Okay, why? Because once any other foreign gene is introduced into a gene sequence, its complete quality will get changed, isn't it? Due to mutation, we learned that, isn't it? So what will happen? Instead of reading that continuously, if you add some other genes in between, the complete amino acid coded by them will be changed, and it all change the quality of the that particular gene. So likewise, now when you add the foreign gene into the BAM H1 site of the tetracycline resistant gene, what will happen? The bacteria, okay, they lose, or the plasmid lose the tetracycline resistance, okay, due to insertion of foreign DNA, but it can be selected from non-recombinant by what? Growing them in a medium containing what? Ampicillin containing medium. If you grow them in a medium containing ampicillin, okay, you can see that ampicillin means that transformants, will survive and the others will not be survived. Understood? The transformants growing on ampicillin containing medium are transferred to medium containing tetracycline. Okay, and the recombinants will grow, means which has already recombined, new genes are added, will grow in ampicillin containing medium but not on that containing tetracycline. Okay, but non-recombinants, means which are not undergone the recombination, will grow on the medium containing both the antibiotics. Okay, so in this, one antibiotic resistant gene is helping to select the transformants, and the other, the other antibiotic resistant gene gets inactivated, isn't it? When the new gene is introduced into the tetracycline resistant gene, that got inactivated and helps in the selection of recombinants. Understood? So in this way, a vector should have a selectable marker, and it should have what? Cloning sites. Okay, cloning sites. So cloning sites means what? Where you can add, and in one, okay, there are two antibiotic resistance genes, and one is getting one is helping in selecting the transformants, and the other is getting inactivated due to insertion of alien DNA, and helps in what? Selection of recombinants. Understood? So like this, what we can select the recombinants from non-recombinants. Clear? Non-transformants. Now, today we have learned about what? The qualities of vectors. Okay, cloning vectors are what? Medium which helps to deliver the gene of our interest into the host cell. Understood? And a vector, usually normal vectors we used are what? Plasmids and bacteriophages. Okay, because they can independently replicate in a host cell without the direction from the the chromosomal DNA. Okay, so what are the characters we were studying? Origin of replication it should have a selectable marker, it should have a cloning site. So all these are very clear to you, I think. We'll continue the rest of the portions in the next class. Thank you.